Protein synthesis. Proteins are the most dynamic and varied class of biomolecules. The singularity of every cell type is owned almost entirely by the proteins it produces. Therefore, it is not surprising that a large amount of cellular energy is used in protein synthesis. Because of this strategic importance in the cellular economy, protein synthesis is a regular process. Although control is also of the fundamental importance at the level of transcription, regulation of the translation of genetic messages allow for other regulatory opportunities. This is especially true in multicellular eukaryotic organisms, whose complex life cycles require diverse regulatory mechanisms.
Protein synthesis is an overall complex process in which the genetic information encoded in nucleic acids translate into the alphabet of the 20 standard amino acids of polypeptide. In addition to translation, the mechanism by which a sequence of nucleic acids directs the polymerization of amino acids, protein synthesis can also be considered to include the process of post-translation, modification, and targeting. Post-translation modifications consist of chemical modifications used by cells to prepare polypeptides for their functional roles. Various modifications aid in targeting, which takes the new synthesis molecules to a specific intercellular or extracellular location.
At least 1,000 different molecules are involved in a protein synthesis. Among the most important are the components of ribosome, large ribonucleic protein, machine that synthesize polypeptides. Each ribosome reads the base sequence of the RNA and driven by GTP rapidly and accurately convert this information into the amino acid sequence of polypeptide. Speed is necessary because organisms must react speedily to every changing environmental condition. For example, the prokaryotes, such as E. coli, 1,000 residual polypeptide is synthesized in less than 6 seconds. Eukaryotes are slower with about 2 residues per second. Accurately in RNA translation is critical because the proper function of each polypeptide depends not only on the primary sequence of the molecule, but also is on the exact folding.
To create proteins, there is a special organelle in the cell called the ribosome. The ribosome is located in the cytoplasm of the cell. However, the genetic code in the DNA is contained in the nucleus, which is another part of the cell. That makes it a bit problematic, since the ribosome needs the genetic code to make proteins. Getting the DNA into the cytoplasm will not be possible. Remember, that is a hug molecule. To solve this problem, the cell has found another simple solution. It creates a small copy or a transcript of the specific gene through the process of transcription. This copy is a small RNA molecule. Can messenger RNAs are very close relatives of DNA. They consist of almost the same material, but while DNA is a double-stranded, RNA is made up of a single strand. RNAs are small enough to exit the nucleus through a small pore in its membrane.
The step in which proteins are actually produced is called translation. It is when the messenger RNA code is translated into small building blocks of proteins, amino acids. Once in the cytoplasm, the ribosome reads the instructions from the messenger RNA. Following the instructions, the ribosome assembles the amino acids in a specific order to form each protein. Each protein is made like a string of pearls, in which the pearls are the amino acids.